Hey guys, Mrs. Horn here. I've got notes for the second half of chapter 35. We're still focusing on the same question, talking about structures that are inside a plant that help it to sustain life. But in the second half, you're going to learn about different types of cells that make up plants, different types of growth that happen in different locations of a plant, and then how that growth can affect the shape of a plant. So picking up where we left off, the first type of cell that I want to mention is a parenchyma cell. Now, parenchyma cells are the least specialized of the cell types that I'm going to talk about. They have a very thin wall. It's a primary wall. These are living cells. There's a picture of parenchyma cells over to the right. Parenchyma cells help to make and store organic products. So that's where photosynthesis is happening. So there must be some chloroplast here. And the organic products that I'm referring to is glucose. Parenchyma cells also make up most of a fruit, so apples, oranges, bananas, fruits that come from trees. If you were to look at the cells under a microscope, they're going to look very similar to the ones in the picture on the right. Parenchyma cells can also differentiate into other cell types. So seeing that they're not very specialized, they can later become other types of cells. So most cells in a plant start off as a parenchyma cell. Collenchyma cells, these are unevenly thick. They also have a primary wall. Their main purpose is to give support for young shoots. So new and young stems of a plant are made of collenchyma cells. These are living cells. They are very flexible, and a great example is if you've ever bitten into a piece of celery and had those long string things kind of awkwardly hanging out the sides, those are collenchyma cells. Sclerenchyma cells is the next type of cell. These are very thick, very strong walls due to the lignin that makes them up. They have both primary and secondary walls. Sclerenchyma cells are found in the areas of the plant where there's no growth. So you're not going to find these on new shoots or new roots. They are dead when mature. They do help give support to the plants. A tracheid and a vessel element, those are types of cells that are made of sclerenchyma cells. Two types are fibers and sclerids. And these are some pictures of sclerenchyma cells right here, the pink area. So now we're moving into the types of growth that a plant undergoes. The meristem cells, which is the region that the cells grow and the region of cell division, mitosis. We also have the apical meristem. This is where this is the growth of the tips of the roots and the shoots. And then the primary growth is where the root cells are going to get longer and longer and start to push through the soil. The elongation of those shoot cells, which are the stems that are trying to head towards the light and get more light and more carbon dioxide. It does occur in both woody and non-woody plants. There's also secondary growth, and the secondary growth happens after the primary growth. And this would be a thickening of the roots and shoots, so adding layers to the roots and the stems to make them hardy. This only happens in woody plants. The product of the lateral meristems is when the dividing cells along the outside of the roots and the shoots produce a new outer tissue, bark. This also helps to add layers of vascular tissue and the xylem. We also know it as wood. The primary growth, now talking about roots. So all of the primary and secondary growth that we just mentioned was growth in stems. Now let's talk about primary growth in the roots. The root cap, which is the structure at the very tip of the root, helps to protect the cells in that highly dividing area, and it helps to push the roots through the soil. In the roots, there are different zones that we have to be very familiar with. There's the zone of cell division. This is where the apical meristem and the primary meristem cells are. We have areas called the protoderm, the procambium, and the ground meristem, and here's information on each of those. The protoderm is the area that will eventually become the epidermis, so that's the outer area. The procambium, this will become the stele. The stele is the vascular tissue where the phloem and the xylem will develop in the root. And then there's the ground meristem, and the ground meristem becomes ground tissue. Another zone is the zone of elongation. This is where cells will elongate up to 10 times their original size, and this helps push the root through the soil. And then the zone of maturation is where the meristem cells will become mature. And there's a good picture at the bottom here. This is where the root cap is, and the yellow area on the screen is the area of the most cell division. This is the area of elongation. You can actually see the cells are looking longer and narrower in size. And then this is the zone of maturation up here, and you can see there's actual root hairs coming out of that cell. In the stems, there is an apical meristem that will give rise to primary meristem. So just as in the roots, stems also have a protoderm, a procambium, and a ground tissue. The vascular bundles, which is made up of xylem and phloem, these are vascular tissue, and they're arranged in groups, and they run through the whole length of the shoot. We saw this earlier in the notes when we looked at the difference between monocots and dicots. We talked about how the vascular bundles were arranged in a monocot and a dicot, and that's what this is talking about. In the leaves, the epidermis of the leaf is the first line of defense against any kind of exterior damage or disease, fungus, parasites. The stomata is a structure that is in the epidermis and allows gas exchange between the atmosphere and the plant. They're little pores. You can find them on the underside of most leaves. It also allows water to evaporate out of a plant due to transpiration. There are guard cells that surround the stomata, and depending on the amount of water that's available, those guard cells will either swell or shrink, which will either open or close the opening. The mesophyll is a layer of ground tissue that's between the upper and lower epidermal cells. And there's a picture of a cross-section of a leaf. This structure down here, these two little purple cells, those are the guard cells. The very small opening, which is hard to even distinguish, there is a small opening between the guard cells, that's the stomata. And the mesophyll would be, if this is the upper epidermis and the lower epidermis, the mesophyll is everything in between. Now, secondary growth is when the vascular cambium produces secondary xylem, which is made up of wood and phloem, and this is what accounts for the increase in the tree's diameter. You know how the tree gets bigger every year and makes more tree rings on the inside? That's what this secondary growth is. There's also a cork cambium, which replaces the epidermis with a thick covering for the shoots and the roots called the periderm. The periderm, the cork, and the cork cambium is actually what make up the bark. And the picture that I have at the bottom here, which is already labeled, shows you those structures just mentioned. Of everything that I just talked about, this chart here is a nice summary of the different types of growth, the different structures in the roots and the shoots. So this summarizes the last few pages. The last section of the notes deals with the shape of the plant and how the growth of the cells and the division of the cells determine the shape of the plant. The direction of the cell division actually determines the body of the plant. If there's asymmetrical division, then the cells are dividing unevenly during mitosis. And the bottom of this picture shows that. So if here's a cell and it divides unevenly, this is what gives a plant different types of cells. The two cells that are the smaller cells look like they're going to become guard cells. The opening in between them is going to be called a stomate. So cells that make up a plant are different sizes and have different functions, and part of the reason why that happens is because of the way they divide, or the way they unevenly divide. And the last page here, about two more terms. Morphogenesis is the development of body form and organization. When you see the word morphogenesis, think shape. And differentiation has the word different in it, so think becoming different. All cells at some point differentiate, even human cells when they're in the embryonic stage, they differentiate to become fetal cells. In plants, all cells in a plant contain the same genes. However, they're not all expressed in every cell. This is the same for all living things. So a plant, every cell in a plant has the exact same DNA, the directions for everything. But a cell that's in the upper epidermis of a plant, it has to function differently from a guard cell, so certain parts of their DNA are actually shut off, while other parts of the DNA are turned on. Cells can make different proteins, which give different structures and functions to different cells in different locations of a plant. This finishes our notes for chapter 35, and I hope this helps.